Welcome to video 3.5. In this video, I'll be covering the secondary structures of protein, starting with the alpha helix. First, let's discuss what secondary structure actually means in the context of protein. If you recall, with DNA and RNA, this had to do with multiple strands of DNA linking together, making helices, and this is a very similar idea of a helix. However, it's important to note that all of the secondary structure we're talking about is for a single polypeptide. One single chain making a helix by itself. So the idea here is to first discuss the primary sequence, which is just the sequence of amino acids in the protein. So for example, alanine, serine, glutamate, aspartate, tryptophan, picking random amino acids. The point is that primary sequence is the instructions. What happens next is the beginning of folding and secondary structure. So secondary structure is when the backbone of protein folds into regular patterns. And so we call this areas of regular structure. Right? These areas here, for example, are helices. Right? We call these alpha helixes. Now, the important distinction here is in between, when there isn't actually a regular area. Sometimes we call this irregular structure. But that doesn't mean that it isn't specific and important. These irregular regions can have really important functions, but they're not part of this regular secondary structure. Overall, proteins that have the secondary structure, for now just talking about alpha helices, right, that is when they get their secondary structure, and tertiary structure is when the whole thing comes together, and we have our alpha helices and areas of irregular structure in between, right? So the tertiary structure is when I'd say the entire thing folds. And it's good to think about it in sequence, that first we make the primary sequence, then the backbone makes these regular folds, right? These patterns like the alpha helix and also the beta sheet, which we'll learn about soon. And then tertiary structure is when the whole thing folds. So this is an example of a protein that was ripped off the internet. And you can see these areas of drawn helixes, right? This is representing parts of the protein structure. They aren't actually shaped like a ribbon, but it's representing these areas where the structure is regular, right? So these alpha helixes are super important. Meanwhile, these spaghetti bits in between can also be pretty important, even though they don't have a regular structure. All of that is part of secondary structure, and when it all folds together into its final shape, that is your protein's complete tertiary structure. So with that in mind, let's dive into the alpha helix a bit more. It is a type of regular secondary structure, and the key idea here is that they're so common and they form spontaneously, they must be stable in some way. So alpha helices are present in not every single protein, but almost all of them. They're very common. And so what makes this form? Why does this form? Right? For DNA, for example, we know that they form because of base complementarity, right? G's and C's meet A's and T's, and this whole thing causes them to interact. But we can't use that for protein. So what's going on, right? The idea is when we look at the peptide backbone, there's lots of hydrogen bonds that could happen, right? Lots of hydrogen bonding. So you'll notice, just looking at the backbone, ignoring the R groups, right? It doesn't matter what those are. We're looking at the backbone. Every amide bond has a hydrogen. That hydrogen could be an H bond donor, right? And by itself in solution, those H bonds, right? If we put this into water, it'll H bond with water. It'll be stable, but it always wants to be H bonded, right? Otherwise, those hydrogens have positive charge. Meanwhile, these carbonyls, they have two H bond donors, H bond acceptors, right? These lone pairs. So ignoring the termini, just looking at the backbone, you can see all the H bonding that goes on here, right, in this orange area. Right, just ignoring the R groups, we see a lot of potential for H bonding. I missed this guy there, and this guy. Point is, lots of H bonding, and we need to make sure that all those H bonds are fulfilled. And you may have guessed it, these regular secondary structures exist because they pair all of the H donors and all of the H acceptors of the backbone. So this is just a little diagram of what the helix looks like, right? Importantly, the R groups are all sticking out. So you have this core backbone area, and then the R groups are sticking out of the helix, right? From a top-down view, we can imagine all the backbone is in this circle, and the R groups are sticking out at different heights. So what does it mean to satisfy all the H bonds? Well, the idea here is that we'd have the NH of one part of our sequence and the carbonyl of another part, and they'd be able to H bond, right? So as we go along, all these nitrogens are able to H bond with different carbonyls, right? I'm not drawing this totally to scale, and it's not so totally important that you know how to draw this at all, right? What's important is that you recognize that all of that H bonding is being satisfied by the alpha helix, right? Instead of needing to find H bonding partners and being unstable, by forming an alpha helix, all of that backbone H bonding gets to be fulfilled. That's why it forms, right? The alpha helix is stable because of H bonding of the backbone. And to be important and a clear thing, it's not the R groups, right? The R groups can matter, but they don't H bond with the helix, right? This helix forms because of backbone H bonding. So key features to pay attention to, it is right-handed, right? Similar to DNA, if we walk down the stairs, if we're imagining this is a staircase, we're constantly going to be turning right. That's how you can tell it's a right-handed helix. And approximately 3.6 amino acids per turn. So what that means is if I imagine, let's say, starting here, and I do one full turn down here, so we're looking at these two, there are about 3.6 amino acids of difference, right? So this could be an alanine, for example, then there's another amino acid, another amino acid, another amino acid. So maybe the third or fourth amino acid are going to H bond with alanine. Right? This can be a useful number sometimes when doing some weird questions. I'm not going to worry too much about it. The important point, and it's typically the fourth residue, is what that says. So one, if this is 1, 2, 3, 4. Right? We'd have, let's get rid of this guy here, the H bonding between these. Right? So you can predict who's going to be H bonding with what in an alpha helix. Now, keeping this in mind, there are different types of alpha helices, but they all share the same thing in common. Right? They all have the same helix with the same H bonding. What changes these kinds of alpha helices is the R groups now. So a hydrophobic alpha helix, you might find this buried in somewhere full of fat, right? For example, the membrane of your cells, right? Very fatty and lipid heavy. So the amino acids you'd find on a hydrophobic alpha helix would be like alanine, right? Things that are nonpolar, phenylalanine, valine, isoleucine, these kind of things, right? These kind of amino acids are nonpolar, they're aliphatic, and so they're going to be attracted to other nonpolar things. That would be a hydrophobic alpha helix, right? A hydrophilic alpha helix is going to be the opposite, right? The R groups, right? The backbone is still stabilized by everything, but the R groups are now going to enjoy contact with water. Things like serine, right? Things like glutamine, maybe some charged things like lysine, right? These amino acids are much more likely to be, they have their own H bonding they need to fulfill, I should say, right? Serine and threonine both have OHs. They have to get their H bonding from somewhere else. They can't get it from the backbone because that's taken up in a helix. So they need to be surrounded by water to be stable, right? So it's really about the R groups. Now, an amphipathic helix is interesting. And you can imagine, right, that we have, as we talked about before, about 4 amino acids per turn. So let's kind of draw some amino acids, right? That means that some of these are going to have R groups sticking out this way, and some will have R groups sticking out this way. And this is, again, all dictated by the primary sequence. What is the sequence of amino acids? That's really where all of this comes from, right? Structure determines function. As this helix forms, perhaps we're going to be putting things like alanine on one side and things like serine on the other. And now what we'd see is that this side of the alpha helix really wants to be in contact with water, and this side of the alpha helix really wants to be in contact with fat, right? That would be an amphipathic helix. And it's the same alpha helix, it just depends on the R groups present. Now, the last thing to talk about with alpha helices before moving on to beta sheets is that proline really plays an interesting factor. Recall that proline is this really weird amino acid that kind of breaks fundamental rules. Instead of an R group connected to an alpha carbon, the alpha carbon swings around and connects to its own nitrogen, making a ring. It's really rigid, and you'll notice that that turning thing we see with the helix, it requires flexibility of the alpha carbon, right? Since there's no flexibility, you can't make an alpha helix. So you'll never ever see a proline in an alpha helix. In fact, what you might see if you're looking at a primary sequence is if the helix is here to here, so we have a bunch of amino acids, let's say there's like 100 amino acids in this helix, there might be a proline as the 101st amino acid. That proline would then stop the alpha helix from continuing because proline is not able to be in alpha helixes due to its rigidity. That's an interesting way that, by primary sequence, by putting proline somewhere, you can control the length of an alpha helix. Or break alpha helices by putting them in the middle, right? If you think about a mutation that mutates an amino acid into proline in the middle of an alpha helix, that would have absolutely destructive effects on the stability of that alpha helix, and it would change the shape of the protein for sure. With that in mind, now that we have a better understanding of secondary structure, let's dive into a slightly more complicated type, and the only other type we need to worry about, the beta sheet, in the next video. I'll see you there.